What's up mga kayuyans? Gusto nyo bang malaman kung paano gumagalaw ang mga molecules sa loob ng inyong katawan? Meron tayong dalawang transport processes. Una, passive transport. Pangalawa, active transport.
Oh, paano? Ready na ba kayong matuto ng tungkol sa passive and active transport?
Tara, matuto tayo!
Katulad sa motorsiklo, sa active transport, hindi basta-basta makakarating ang mga molecules kung walang kulong. Kailangan nila ng gasolina o ATP bilang energy. Dahil dito na ipapasa ng cells ang mga molecules mula sa mababa hanggang sa mataas na concentration.
Walang passive transport ay paraan ng paggalaw ng mga bagay gaya ng tubig at hangin papunta sa high to low concentration na hindi na nangangailangan ng enerhiya to reach its equilibrium.
Eggs-Periment.
Ganyan ang diffusion, isang uri ng passive transport na kung saan gumagalaw ang mga molecules from high concentration to low concentration, katulad ng pabango. Mula sa bote na mayroong mataas na concentration, kapag binuksan natin ito, gagalaw ang mga molecules papunta sa malawak na area, parang paggalaw ng mga molecules papunta sa low concentration.
Narinig niyo na ba ang term na osmosis? Ito ay isang proseso kung saan ang water molecules ay nagmo-move palabas o papasok ng cell para mabalance ang concentration nito sa pagitan ng ating cells at outside environment, na siyang nagpapatibay sa mga halaman tuwing nagdidilig tayo. Para bang siso, kung may 4 na bata o water molecules sa kanan at 2 lang sa kaliwa, di ba hindi balance? So, may isa na lilipat sa kabila para mag-balance o equilibrium at makapaglaro sila. Ang osmosis ay may 3 uri: hypertonic, isotonic, at hypotonic.
Sa isang isotonic solution, ang solute concentration sa labas ng cell ay kaparehas ng solute concentration sa loob nito. Walang water movement na mangyayari sa plasma membrane dahil sila ay nasa equilibrium na.
Kapag naman sinabi nating hypertonic solution, ibig sabihin mas mataas ang concentration ng solute sa labas ng cell compared sa loob. Dahil dito, water molecules will come out of the cell in order to balance the concentration. At ang resulta, magsishrink ang cell dahil wala na tong laman na tubig sa loob.
Sa hypotonic solution naman ay mas mababa ang concentration ng solute sa labas kumpara sa loob.
Dahil dito, may mga pumapasok na tubig sa cell na nagdudulot ng pamamaga at pagsabog nito.
But wait, wanna know what happened in our experiment? Watch this.
Fill the beaker with cold and hot water. Measure and record its temperature. Secure it with a piece of cloth with rubber band. Put a few drops of food coloring into the chamber and observe. Record your observations.
Using a straw, poke a hole in the gelatin, then add a drop of food coloring. Measure the diameter of food coloring as it diffuses in the gelatin.
Carefully measure the egg's mass, put it in a transparent jar, then fill it with vinegar. Record your observation for 5 consecutive days.
The dye diffuses fast in hot water and slow in cold water because higher temperatures increase the molecular movement, speeding up diffusion.
Water continued to move into the eggs in the hypotonic solution of salt water, further increasing their mass. Eggshells contain calcium carbonate which reacts with acetic acid and vinegar, dissolving the eggshell. This reaction forms acetate and carbonic acid. Carbonic acid is unstable and eventually splits into water and carbon dioxide that we see as bubbles around the egg. After 24 hours in each solution, the mass of the egg changed because of osmosis. When the eggs were placed in vinegar, the shell dissolved and the eggs gained a large amount of mass since the solution was relatively hypotonic, causing water to move into the eggs.
In the arnibal or sugar solution, which is strongly hypertonic, a greater osmotic gradient pulled the water out of the eggs rapidly, significantly decreasing their mass and making the eggs appear shrunken. Due to tap water being hypotonic relative to the eggs, water entered the membranes once again, further increasing their mass, eventually causing the eggs to burst due to osmotic pressure from its environment. In hypotonic solutions, the egg gains water and becomes heavier, while in hypertonic solution, it loses water and becomes lighter. The egg gained mass in hypotonic solution like vinegar and tap water as water moved inside, and lost mass in hypertonic solutions like salt water and arnibal as water moved out. The bursting happened due to excessive water influx creating high internal pressure.
Ay, pag nagbukaan na.